in black. I'm going to go to the example sequences here. These are sequences generated by students. They're being opened in Chromis. There are a couple of other software, so I'm going to open the sequence. It's already open, it looks like. You can see here that here's standard Sanger dideoxy sequencing chromatograph, and I've already trimmed off the poor sequence. There may be some other issues here that I didn't trim off. I want you to note that this sequence I picked, it's actually heterozygous at this position. So one parent has a T, one parent has a C at this position. And I've actually highlighted the coding sequence here. So I've already actually done an option here where I'm showing the translation. So let me go back. This is the default view. Under options, you can say I want to see the translations. And if you don't know what frame it's in, whether it starts on the first, second, or third, you can hit 3 lines. And the top will be starting on the first base called, starting on the second base called, and starting on the third base called. I happen to know that this is in 3rd frame, so I'm going to go back and show the translation in frame 3 only. And you can see here that you've got the normal amino acid sequence of the gene actinin 3 with a stop here. If this were a C, this would be CGA, this would be arginine, hence the name R577X, or arginine at position 577, or a stop codon. So that's the change here. We're going to blast this sequence, but before we do that, I'm going to do a quick, I'm going to copy the sequence so I can use it for something, and I'm going to copy it fast to format like that. Then I'm going to go back and blast this just to make sure this is actually a human sequence. And if you hit blast, it'll take you and submit a job directly to the database. So it's talking to your browser. Hit OK here. You're doing it against just nucleotides. You don't need to change anything here. It's going to kick you over to the blast tool. And it's going to sit here and queue this up and run the blast. And you can hit this and see how fast it operates. Whether it's done, it finished very quickly here. And you can see there are a couple hits where it aligned almost perfectly all 310 some odd bases. If I scroll down, you'll see that it aligned to a clone somebody uploaded that has been determined to be on chromosome 11, and then do a reference sequence, which is more important here. Let's look at this clone. You can see that this is our sequence, and here's our TGA. This is the stop codon. It turns out that the clone that was uploaded was also had a T at that position, or would have been a premature stop in the actinin 3 gene. If you look at the actual reference sequence gene, which is the curated gene that's known on chromosome 11, so this has been professionally looked at, there is probably a sequencing error about 8 bases in. You'll see this is our arginine to our sequence is a TGA, which would be a stop codon, and here is the CGA, which is the normal or reference sequence, hence the reference sequence gene. So this is a good way to show the students that the sequence is indeed human. So now we're going to go over to the Genome browser at Santa Cruz, genome.ucsc.edu, and we're going to use a tool called BLAST, which is a BLAST-like alignment tool. So, but rather than going to a database, this aligns to a genome. It does ask you to know what genome you're working with. We're doing human here, and this is the most recent build. I'm going to copy and paste my sequence in here, and just for the purposes, I'm going to do test actinin 3 and submit it. This actually works fairly fast. It's a small fragment, and this puts the sequence in the context of the genome. So you can see here, these are our sequences, the tests. I've done this before, and it remembers that I've done this. So you can see here, these are tracks or linear representations of the genome. I'm going to zoom out tenfold. A way to show students that your PCR product is in the context of introns and exons. In this case, the gene codes, I believe, in this direction. So this is exon, whoops, didn't mean to do that. If you mouse over, this is exon 15 and exon 16. So we actually PCR'd and then sequenced a piece of exon 15 on into the intron 15. Please note that this red line actually is significant, and that's the likely variant. It looks like there may be a variant out here in the intronic sequences, which is probably not relevant. I'm going to quickly zoom into base, which is at the nucleotide base level. So it's just zoomed down into our black bar here. And you can grab this and scroll, I believe, or move back and forth with these buttons. But importantly, you can see here, this is the amino acid sequence of the gene. And there's the arginine. R is coding for arginine. The reference sequence is C, G, A, and our sequence has a T. We know that this actually is a variant. It's highlighting that this is an allelic variant in the online Mendelian Inheritance of Man database. So we could look up this variant in OMIM if we wanted to spend the time. And also down here, you can see that the database picks up. This is a SNP number. So you can see if you highlight this, it's going to take you out to DB-SNP, which is a database of all human SNPs. It's one base pair long. It gives you the exact location. It shows you that the reference allele is C, that chimp, orang, and macaque all have C, and the variant is T. The other thing this also gives you is frequency data. So here's the frequency data. 55% of known sequences have a C, with T at 44, and this is how they get the percentages. And so there are 125,000 known sequences of this gene for human, and that's the human variant. Let's go back. So this, I hope, takes a sort of abstract element on a gel, sequences it, and then drills it down to the genome level. Okay, so there's another way to look at it, which means we can do it in a genome database called Ensemble. I'm just going to go to Ensemble here, go to home of Ensemble, show you how to do this. You go to home of Ensemble, you have to pick your genome, but we're going to do the same thing and go to BLAST/BLAT and do a new job. And again, we're going to put in our actinin 3 sequence, and it's DNA, and we want to do this as a BLAT for the first time. We're going to run this. I've obviously done this a few times, so trying to get this recording to work right, but let's look at the results here. This usually only takes 30 seconds to update. And you can see here, this is giving you the context in this first screen. So you can see it hits, the best hit is on chromosome 11 again. Gene is actinin 3, 306 base pairs aligned. Now 23 base pairs aligned, 23 base pairs aligned on chromosome 1, but 23 is probably in the intronic sequence. So the native sequence is here on 11. Look at the genomic region here. And this is just putting the same kind of style here, showing the exact where we were. Here's actinin 3. And here's the DNA sequence that we generated. You can see it says TtGG, so let's put it in the context. And these, again, are the SNPs. You can see, you can mouse over these and highlight them, and it'll tell you what the SNP is. The alleles are C and T. It's a stopped gained. You can look at population genetics, which is nice from here, which is a different view than the frequency view we saw before. See if it'll work. There we go. You can look at frequency in different populations. It's loading down here. And these are subpopulations, all humans, Africans, American subpopulations. So Finnish is different than British versus Iberian and Spain versus Italian and so on. Okay, let's go back. I think the other great value of Ensemble is its ability to look at gene trees. So I'm going to go to the gene now. And the gene, you have to be in the gene tab, not in the results or not in the location tab, and you can go to the gene tree. The gene tree shows you all it builds a phylogenetic tree or evolutionary reconstruction. And right here in the middle, you see the human actinin 3, that's our sequence, and you can see its nearest neighbor is gorilla. Here's chimpanzee. I'm not sure why gibbon and gorilla. I'm not sure why chimpanzee isn't showing up. And also, you can expand these to look at mouse, for example. So the next step is understanding, does this sequence or does this gene lend a function? And the way to look at that is to go to the mouse genome database and see if anybody has knocked this out. So I'm going to go to mouse genome, and you can see that comes up as mouse genome informatics. And here we're not going to use the sequence, but we're going to look for the gene. So I'm going to go to the gene page and go to genes and markers query. And I'll put in here the name of the gene, ACTN3. And in mouse, it's on chromosome 19. And this will tell us a little bit about the gene, alpha-actinin 3, what its human ortholog is, which we know, and you can see what its phenotypic overview is. It's involved in homeostasis and muscle physiology. It also tells you that there's actually been some work on making alleles, either trapped or mutations. So there are mutations in the mouse example too. Rather than go through all of the mouse database, I'm going to go to these two targeted or knockout genes. And you can see that this gene has been targeted and knocked out twice, once in a cell line and once in a mouse. And if I hit this, it'll take me to their gene report. So this team, MacArthur et al., knocked this gene out. And if we want to know if they learned anything, we can read the abstract. So here's the abstract, and I would just want to highlight that they show that actinin 3 expression in the knockout shifts muscle metabolism toward the more efficient aerobic pathway and increases and causes an increase in intrinsic endurance performance. If you read this paper, you'll note that they put knockout mice who are otherwise normal on a treadmill and looked at their performance. So this is a nice way to go from sequence to prove it's human with BLAST, to use BLAST to put it in genomic context, and finally to go to MGI, their mouse genome database, to look at function.